Okay, 10 minutes. So today I'm going to tell you about a study where we've used 3D liver microtissues in conjunction with novel micro-TMA technology to make cross-species comparison of CAR-mediated key events in liver carcinogenesis. So the incentive for using 3D microtissues in this study was that they may make a more representative model than 2D hepatocytes because they behave more like hepatocytes in vivo. The hypothesis that we tested in this experiment was that these 3D microtissues could recapitulate key events in the process of CAR-mediated liver carcinogenesis, enough to support a mode of action at that level, underpinned by Bradford Hill considerations of causality. And our vision for this approach would be to apply this earlier in the discovery process to identify compounds that have this liability, and so that you could, once identifying these compounds, you could reduce the associated attritional costs caused by these adverse events occurring, and I will discuss this a bit later on in the talk. Okay, so liver tumours involving activation of constitutive androstenedione receptor are a common occurrence in 2-year bioassay tests, and this mechanism is not considered relevant to humans. And there are 5 key events necessary for establishing a CAR mode of action for liver carcinogenesis, namely CAR activation, altered gene expression, secondary to CAR activation, increased hepatocellular proliferation, clonal expansion, and ultimately hepatocellular tumours. So today I hope to convince you that 3D liver microtissues coupled with this micro-TMA and genomics analysis can recapitulate 3 of these 5 key events. And we've used micro-TMA-based quantitative histopathology to measure hepatocyte proliferation in these 3D models, and also transcriptomics integrated with proteomics to assess CAR activation and altered gene expression in parallel samples. So the micro-TMA technology works by placing liver microtissues in a genomic grid, retaining the 96-well experimental layout. And this grid is then embedded in wax and sectioned onto microscope slides. And we can make up to 20 sections through all the spheroids in the grid, which provides a very high capacity for multiplexing. And for example, we've recently developed a method for transcriptomics on laser dissected spheroids from the micro-TMA by placing the sections onto membrane slides, so that we can do quantitative histopathology and transcriptomics on parallel sections. So the present experiment, we used rat and human liver microtissues exposed to the prototypical CAR activator phenobarbital over a range of concentrations and times. And BRDU was added to the treatments 24 hours before harvesting the microtissues. The tissues were fixed in paraformaldehyde before being analysed by immunofluorescence for BRDU labelling index. So this shows an H&E map of one of these micro-TMAs, and we exposed the tissues to different concentrations of phenobarbital over a range of times, and also exposed them to a positive control, which was hepatocyte growth factor. Now, the power of the micro-TMA is really to be able to generate multiple biological replicates for any particular treatment and stain them all at the same time. And this reduces technical variability, which facilitates quantitative histopathology. So we employed 3 different staining techniques on parallel sections in the micro-TMA. The left panel shows CD68 staining in these primary liver hepatocytes, primary liver spheroids, to identify Kupffer cells. And the middle panel shows a CD31 staining to identify endothelial cells. And the right panel shows BRDU immunofluorescence staining, which shows up in red in this case. And the green stain is cytox green nuclear counter stain. So there are, if you look at the right panel, there are BRDU positive hepatocytes and also non-parenchymal cells, shown by the red and yellow arrows. So using this staining methodology as a guide, we were able to design an image analysis algorithm that automatically counted BRDU positive hepatocytes across the TMA. So this shows the image analysis process to derive a BRDU labeling index using this automated script. And the image analysis is performed in 4 steps. Firstly, the script uses a difference of Gaussian filter in the cytox green channel to identify non-parenchymal cells and saves the coordinates of these cells. And next, the same or similar approach is used to find hepatocytes, where the nuclei are bigger and rounder than the non-parenchymal cells, and the coordinates of them are saved. And in step 3, the algorithm thresholds in the red channel a given distance from the center of the nucleus to identify BRDU labeled hepatocytes, and finally to calculate a labeling index for hepatocytes in the spheroid across the TMA. So this shows examples of images from these different treatments in both the rat and human liver microtissues, the rat at the top and the liver in the third panel from the top. And the histograms show proliferation data expressed as hepatocyte labeling index for both rat and human. And we found that the rat hepatocytes proliferate in response to phenobarb in a dose and time-dependent fashion, whereas those of the human liver do not. And this was the expected result, as it mirrors data previously published in 2D hepatocytes. However, the difference in this case is that the response is prolonged over a much longer time period. 2D hepatocytes lose the ability to respond in this way after approximately 36 hours. So we used a proprietary method also to obtain data on differentially expressed CAR-regulated genes using expression microarray, and this was performed on parallel samples, and for each data point we used 2 to 4 microtissues. And this shows data for CAR-regulated phase 1 and phase 2 enzymes, the rat in the top panel and the human in the bottom panel. And we observed a dose and time-dependent induction of these phase 1, phase 2 CAR-regulated genes, indicating the CAR receptor was functional in both the rat and the human microtissues. Again, using a proprietary proteomics method in these fixed microtissues, we also obtained data on protein expression, and this was integrated with the microarray data. And this table shows that data for rat in the left and human in the right panel. And if you look at the columns highlighted in yellow, this shows the protein data, and we're able to show that for many of the genes that were upregulated in this case, in this CAR-regulated pathway, they were also upregulated at the protein level. Okay, so in summary, we've used a micro-TMA-based quantitative histopathology technique to show that rat hepatocytes in 3D microtissues proliferate in response to CAR activators, in this case the prototypical CAR activator phenobarb, but humans hepatocytes do not. And using integrated transcriptomics, we've shown that phase 1 and phase 2 enzymes regulated by CAR are induced at the RNA and protein level, showing that CAR is functional. Lastly, we've used Ingenuity upstream regulator analysis to indirectly demonstrate activation of the CAR receptor in this model. So this is the first time this has been done, and this data was recently published in Toxicology Reports. And I think one of the strengths of this type of assay is its ability not only to examine and consolidate a mode of action, but also in the same assay to be able to assess the human relevance of this pathway. So more recently, we've adapted this micro-TMA technology to be able to perform toxicogenomics in parallel, and done this by laser dissecting the individual spheroids out of the micro-TMA by sectioning the micro-TMA onto membrane slides. And this can be automated using this robo-mover technology, which is shown in the bottom two panels. And the way it works is to automatically catapult each spheroid into lysis buffer in a 96-well format. So we've been able to use this in conjunction with a new NGS-based transcript profiling system called TempoSeq, and this work is being done in collaboration with BioClavis, and I think there's a BioClavis poster here, and so we're working with them on that. And our vision really for this approach is to use the platform prospectively earlier in the discovery process to identify compounds that have this toxic liability. And this principle has been illustrated by this phenobarbital study that we've shown, but it will be possible to apply this more broadly by doing this automated genomics and phenotypic analysis, and then comparing these transcriptomic signatures against a database of adverse outcome pathway signatures, so that we could identify toxic liability with compounds earlier in discovery. And this would reduce the attritional costs associated with that later on in the process. So just to finish then, we have a poster here which talks about this technology, which is quite flexible actually. We've applied it to many different 3D organoid disease models. So if you're interested to find out a bit more about how this can be used to assess these spheroid and organoid models, please come along and talk to us about that at the poster. Okay, thank you.
Thanks very much. You mentioned that you can do this experiment with different biological replicates. Could you define what you would classify as a biological replicate, please?
I think the question was, did we do a statistical power analysis to assess how many biological replicates we needed to show the effect? Maybe not.
It was just to define whether what you classified as a biological replicate, these separate donors, and then subsequently.
Okay.
What
we had to answer that question, I know, it was part of the question that was asked for the paper. It's a very good question. So, the way these spheroids are used is, in this particular case, these are commercially available spheroids, but it would be applicable for your own spheroids that you're working with. They're incubated, in this particular case, in gravity trap plates, so that the incubation for each one of them is a separate entity, effectively. They're being exposed individually, so they can be considered biological replicates. I mean, I guess you could draw a parallel, probably a leap of faith between each spheroid effectively being an individual animal, if you wanted to draw a parallel from animal type studies.
Do they come from different individuals?
Right. No, I mean...
then can you really consider the biological replicates?
That's getting at the question. Well, okay, so I'm talking about biological replicates at the level of treatment, not biological replicates at the level of the human population. These were spheroids derived from mixed primary liver resections from a number of humans, I think 6 different humans. So they're not biological replicates at the population level, but they are biological replicates at the treatment level. And I think the point about this really is quite practical, really. I mean, what we're trying to do here is to design and build assays that are fit for purpose to be able to assess complex toxicological responses that occur over a long period of time. And so it depends on what you're trying to do. The question you're asking is very important, and it turns out that that's a study that we're currently planning. You can buy these primary human microtissues from individuals, and they are available, and so the question of biological variability in the human population is very important. And in Sphero, it's not gone unnoticed to them that there's a lot of fatty liver disease prevalent in populations that's growing all the time, and this clearly would affect human biological responses to drugs. And so they have developed, and we've been working on a NASH model based in these primary human liver spheroids.
Do these spheroids develop viral nuptials? And if they did, could you use them to look at mechanisms of cholestatic toxicity?
Yes, I mean they don't reorganize themselves in such an elegant way as you can achieve using stem cell-based models, where the cells do actually make structures that look like liver lobules. However, they do reorganize themselves sufficiently to produce bile duct-like structures, and there are various assays that have been applied in this way to look at transported bile into bile ducts, and other methods for staining, fluorescent staining, to assess the potential for cholestasis in this model. How representative that is of cholestasis in vivo is open to question, I think, considering the differences really in similarities between the structures are not very good, I think, or not as good as you might want them to be. And I guess we may need to wait for stem cell-based models where these structures are actually formed in a more sort of in a way that's controlled by ways in which only stem cells understand. And these are the kind of miracles that happen, when you're working with these organoid-like models. They do organize themselves in a very elegant way.
You talk about sort of long-term effects. How long can the organoids be used for reasonably and get an effect?
Well, that's the strength really of using these microtissues, and there's a nice paper actually preceded the work that we did by Messner, showing that these responses can be maintained for up to 90 days in these models. So it is possible to use them to model these more chronic effects, which are more realistically parallel or translatable to the in vivo situation. And that's really the strength of these organoid and 3D type models. Unlike the 2D hepatocyte models, they retain this machinery for regulating this CAR-mediated response and also other responses for a much longer period of time. They maintain their phase 1 and phase 2 enzyme levels, for example. So they're useful also for metabolic studies that can be done in conjunction with these kind of phenotypic studies.
The reason I asked the question was there's a bit of recent reading where the same claims were made about stem cell-derived multicellular liver models, about liver slices, and they also said that you keep a liver slice for multiple weeks. They can do multicellular stem cell models for several months. So I'm just trying to compare this advantages.
Yeah, no, they have the exact, the comparison between these and organoid models. They're different from organoid models, but you can maintain them in culture for many weeks, and they do retain this. So, albeit they perhaps, from a histological, structural point of view, they're not as, they don't resemble the human tissues as well as the organoid models. They do survive for a long period of time, and so it's a useful model to be able to assess those chronic type toxicities.
Any further questions? Thank you very much. I'd like to thank the speakers we've had this afternoon.